Good morning, everybody. I'm so happy to be here and present really one of the most exciting topics, especially, given the pandemic, seeing how nanoparticle-based solutions can overcome such a devastating disease. So actually, nucleic acid therapeutics are unique types of drugs that contain biological instructions, genetic instructions, really very different from conventional therapies. Think about cancer. We used to focus on chemotherapy or cytotoxic drugs that had to kill every cancer cell. Now we can actually leverage the immune system and construct it in order to help us actually identify and eliminate those cancer cells. So there's actually a broad spectrum of diseases and types of modifications that we can use nucleic acid for, especially for gene silencing, altered expression of genes as necessary, and actually outright gene editing using CRISPR. And there are two main categories for delivery systems, as you know. Viral vectors that have been used for many years. They really have high transfection efficiency and natural tropism to tissues and cells. However, concerns about packaging, manufacturing, and immunogenicity resulted in investment in research effort for the development of non-viral delivery systems. Those systems contain many types of particles that can be made of polymers, lipids, and can be engineered to deliver actually any type of nucleic acid. We can have actually high packaging efficiency, ease of manufacturing, and reduce toxicity by engineering of those platforms. So nucleic acids really are very powerful, but they're not designed to effectively be delivered into cells. And that's why they're prevented from entering many of the cells of interest because of many biological and chemical barriers that exist. So actually, they need a delivery system. They don't know where to go. So non-viral materials, such as lipid nanoparticles, can provide with protection, improved internalization into the cell, and then intracellular delivery into the right compartment in order to execute their action. Taking lipid nanoparticles, for example, that are now known very well, they are biocompatible nanoparticles. They can deliver many types of genetic materials, and they can protect the cargo from degradation, facilitate their uptake into the cells, and then releasing this cargo. But on the right side, you can also see that we can actively target those particles to areas of interest by coating it with different materials in order to tailor the protein corona and then the distribution of the particles, by conjugating antibodies, ligands that will drive them to wherever they need to go, or different polymers that will alter their biodistribution by changing their physical and chemical properties. So probably the most famous example are the lipid nanoparticles for COVID vaccine. And they really combine the best of both worlds, a nucleic acid delivery system in the form of mRNA that is programmable, can result in transient expression of proteins, and doesn't require genomic integration, together with lipid nanoparticles that can condense mRNA of interest, enhance the uptake, and are relatively easy to manufacture. So with that, those nanoparticles can help drive a particular mRNA into the cell. In the case of COVID-19, it's the spike protein that is presented on the surface of the virus, and the mRNA will be in charge of producing this protein, which will result in multifaceted immune response and activation of T cells, CD4 and CD8 positive T cells, B cells for the formation of antibodies and immune memory. And that's why we see that this trained immune system is now primed to identify and eliminate this virus upon infection. Another successful example of implementing non-viral delivery system is for Transtartine Amyloidosis, where people with a mutant TTR gene create TTR that then assembles and forms those fibrils that get deposited in different organs, resulting in cardiomyopathy and neuropathy, among other symptoms. So the idea was, can we use lipid nanoparticles with siRNA that will silence this target, degrade this mRNA of the TTR protein, which will result in less of protein translation. So that's exactly what this drug, patisiran, is doing. It binds to the wild type and mutant TTR RNA transcripts, degrade them, and we have less available for translation. Indeed, this leads to less of the TTR protein in the serum of patients with this disease, which reduces the formation of amyloid fibrils and the organ damage that is seen in this patient population. So this is really exciting. There are several types of gene therapies that are clinically approved using oligonucleotides, CAR-Ts, and using AAV for their delivery. But now we see the emergence of new opportunities using non-viral delivery systems that can be used for different disease indications and different types of gene therapy, including gene editing, as well as personalized therapy and epigenome editing as well. So thinking is also shifting right now. When we were targeting particular cells depending on the disease and the pathogenesis of the disease, for example, take cancer, really looking at chemotherapy to kill every cancer cell, now the thinking is shifting into immune therapy. How can we leverage our immune system to naturally attack the invader? So with that, we need to think about what we are targeting in terms of organs and cells. Now, instead of cancer cells, for example, we want to also target macrophages, natural killer cells, and dendritic cells. And all of these can be actually done with nanoparticles as well. We can leverage nanoparticles that were designed to target cancer cells, for example, to now target immune cells in lymphoid organs and to deliver this cargo under a particular pattern of interest in order to enhance the outcomes while reducing side effects and toxicities that are seen with systemic delivery of free drugs. So another important consideration, as I hinted, is the role of administration route, because this will affect where the therapy is going in terms of the organ and the cell of interest, the biodistribution and the stability. And we can think about what type of therapy and material will be optimal in order to achieve therapeutic benefit. So rational design of materials, along with utilizing what biology provides us with, can help us iterate more effective therapies. So to date, most of the materials are designed to just be released, go into cells, and perform their action. But we were asking, can we actually design materials that can respond to the environment and leverage some signals or cues that are relevant to disease in order to release the cargo in an active manner? So here you can see an example of self-assembled nanoparticles that we designed for nucleic acid delivery. On the left, you can see two types of microRNAs that we assembled to form triple helix. One microRNA is a mature microRNA that is actually eliminating cancer cells, and the other microRNA is an antagomer that is silencing an RNA that is supporting tumor growth. In order to deliver both of them effectively at the same time, we formed this stable triple helix structure that's self-assembled with a dendrimer. This is a nanoparticle that is multivalent, and we can have multiple copies of this microRNA triple helix interacting based on electrical charge between the positively charged particle and the negatively charged microRNAs. So we now have self-assembled nanoparticles that are really good at entering into cells. This dendrimer, by the way, can interact with another polymer, dextran, to form a gel. So here what we've done is we have a gel that is formed in situ. It degrades over time, releasing those nanoparticles, the dendrimer with the triple helix, that go inside the cell. And only inside the cancer cell, the microRNA will be disassembled and will perform its action. Another example is a nanoswitch for gene therapy and on-demand drug delivery. Here, what we wanted to think about is how can we overcome the problem of multiple drug resistance that is developed for chemotherapy, that more than 70% of the patients develop over time following chemotherapy administration. So here what you can see is a design of a gold nanoparticle that is conjugated to a nanobeacon or antisense DNA with a drug chemotherapy intercalated into the stem of this nanobeacon. Now, this drug will only be released inside the cell upon opening of this hairpin DNA when it identifies a target mRNA. Here, the sequence was designed to interact with multi-resistant protein 1. It's a protein that is responsible for the development of resistance. And the idea was only when the particle goes inside the cell, this hairpin DNA will open and hybridize with the target mRNA of this MRP1 protein, preventing its translation, basically minimizing the development of resistance. And only at that point, the chemotherapy will be released, so it can be active. So this is a design of an active or triggered release of drug based on information that exists in the cancer cell. But we wanted also to get a report on all of these events, to get a signal sent to the outside world. So we used fluorescent imaging for that. So what we've done is the drug here in red, this face inside the nanobeacon, is fluorescently labeled, but its signal is actually quenched by the gold nanoparticle. And the antisense DNA is also labeled. You can see here donor 1, but it's quenched by quencher 1 that is also conjugated to the particle. So in this configuration, this structure is optically silent. When it goes into the cell and the nanobeacon opens up, now the fluorophore is really far away from the quencher, restoring its signal, and then the drug is released, again, restoring its signal. So we can get actually a report on the opening and binding to mRNA, as well as the release of the drug. And this, in animal models, showed very good efficacy and elimination of resistance to the drug. So now I want to illustrate some other technologies that we can use along with nanoparticle non-viral delivery systems to facilitate where the particles would go. So if we think about tumors, in many cases, it would be really easy to apply drug intratumorally. We don't have to be worried about a lot of the biological barriers that are associated with systemic delivery. But it's not very practical in many cases, and not all the tumors are accessible. So in my lab, we developed nanoparticles that are stable in the serum, can be delivered intravenously, as well as the delivery of transdermal delivery patches, which you can see here on the bottom left. It's a microneedle patch that we can deliver drugs directly into the skin. And local delivery platforms. This is the sort of hydrogels or adhesives that I mentioned that can be injected or delivered directly to the target site. They can be programmed to degrade over time and release drugs sustainably. So I'll give you 3 quick examples why this is important to combine different biomaterial platforms with non-viral delivery platforms. So here, we use the hydrogel, adhesive hydrogel for delivery of therapeutics in the brain in glioblastoma. And why are we interested in that? So in fact, of all organs, really drug delivery to the brain is particularly challenging because all therapy molecules have to cross a very selective membrane, the blood-brain barrier, that is designed to protect the brain from invaders such as viruses and bacteria. And so in fact, 98% of small molecules cannot even penetrate the brain. So our therapies that could have been very active do not get to the target site. So the idea was, can we actually bring the therapy to exactly where it needs to go? This is a model brain, and following tumor resection, we can inject 2-component polymers that upon mixing immediately gel on demand and adhere to the tissue, forming adhesive bonds. This adhesive can deliver the drug locally exactly where it's needed, so we're not worried about crossing the blood-brain barrier, allowing us to now deliver any therapy of interest and release it over time. So you can see here mice that we injected with brain cancer cells that develop a tumor. You can see for untreated group, the tumor grows within 12 days. You can see a strong signal. When we treat those mice with oral temozolomide, the current clinically used chemotherapy, you see that actually we don't see very good benefit even in mice. which explains the low survival benefit that we see in patients. And this is because the lack of penetration or effective penetration to the brain and the use of not very potent drug, because imagine that 99.9% of this drug is actually circulating in our body. If it's very potent, it can be very toxic. So with our technology, we identified a more potent drug that now we can deliver because it's delivered locally, and you can see how nicely the tumor is being eliminated. So we wanted to see how this translates into survival of the mice. So here you can see the survival of mice that were injected with a brain cancer tumor is about 22 days. When we deliver the current chemotherapy temozolomide orally, we don't see much benefit. But interestingly enough, when we deliver a much more potent drug directly to the tumor in a free form, intratumorally, we don't see benefit either. And this is because of rapid clearance or elimination of drugs in the brain. When we take this same drug and now deliver it using our adhesive hydrogel platform, enabling local and sustained delivery of the drug, we see now 60% of the mice were able to survive. And now that the tumor growth is attenuated, upon combination with the systemic temozolomide, we get improvement in survival. In fact, we get 80% curative survival in combination with the standard of care chemotherapy when we deliver a potent drug right where it's needed. Brain cancer tumors, they don't metastasize usually from the brain, but they do tend to recur locally. So we were asking, what will happen to these cured mice upon re-challenge when we inject them with another tumor? Would they survive because they generated maybe immune memory that was induced by our therapy or not? So in the absence of additional therapy, we now re-challenge all of these cured mice with an additional tumor. And what was nice to see, it was that all the mice were able to reject the tumor. We've seen 100% curative survival for more than 1 year now. So in fact, our therapy was able to not only eliminate the remaining cancer cells, but also use them to educate the immune system to identify them and eliminate them wherever they are. So in the absence of additional therapies, we were able, the mice were able to reject the tumor completely. In another example, I would like to show you how we can use a microneedle patch for transdermal delivery that enables a very effective therapy delivery as well as diagnosis by sampling of biomarkers in interstitial fluid in our skin. This can report on the health state of the patient as well as the response to a therapy. So this is a microneedle patch. The idea is to apply to the skin like a Band-Aid. It can deliver the drug, and after the drug has been delivered, we can now remove this Band-Aid and analyze biomarkers. And those could be soluble factors, such as proteins, cytokines, as well as cells, immune cells, for example, that give us a lot of information, again, in response to our therapy. So we were thinking, how can we develop microneedles that can both deliver and diagnose? So there are many criteria that need to be achieved here. There needs to be high mechanical strength to actually pierce the skin, the ability to release drug on demand, and also for the materials to swell and sample as much as we can interstitial fluid. In mice, it's only 5 to 10 microliter. So that was important to be able to extract as much as we can so we have enough sensitivity to detect changes. So the idea is to, in the case of cancer, for example, we can deliver immune stimulators, such as TLR9 agonists, deliver it in order to suppress tumor growth, and then remove the patch in order to surveil for the immune response to the therapy. And this is very relevant as we think about new and emerging immunotherapy approaches. So the idea was to use TLR9 agonist or CpG. We complexed this CpG with a polybeta amino ester nanoparticle. This is a FDA-approved and biocompatible particle that can complex with a lot of the nucleic acids. We can deliver this in order to modulate the immune environment in the tumor area and then remove it to assess biomarkers in the skin. So we could see that indeed, if we look at the tumor volume over time, when we deliver the CpG using the microneedles, we attenuate tumor growth, which results also on the right on improved survival of these mice that receive the therapy compared to the untreated mice. When we look at the tumor biopsy and characterize its microenvironment, we can see on the left reduced cellular proliferation after the treatment, and on the right, evidence for activation of both dendritic cells and macrophages. And when we look at what we see in the interstitial fluid from the microneedles, we see a very similar response. With the microneedles, we could see increase of CD8 versus CD4 T cells and more dendritic cell infiltration as well as macrophage infiltration. So even in a very small volume of interstitial fluid that we can extract using microneedles, we could get all of this information, eliminating the need to get a biopsy from the tumor. This allows us also to get temporal information. Because microneedles can be applied in a non-invasive manner, you can apply and remove multiple times. You can get the temporal response to the therapy here, as you can see, over time. And in the last example, I would like to show you the results on the synthesis of nanoparticles for the delivery of a very important immune adjuvant STING, or stimulator of interferon gene, that STING agonist that is now under clinical trials for cancer therapy. So STING agonist, or CDN, as you can see here, is a cyclic dinucleotide and has huge potential as an immune adjuvant for cancer therapy. It actually acts as a pump molecule, a molecule that represents viruses and sequences from viruses and bacteria, and hence activate the innate immune system. STING agonist, when it gets into the cell, interacts with the STING receptor, activates interferon regulatory factor 3, and results in the release of type 1 interferon and activation of many immune cells. So the STING actually is being uptaken by dendritic cells. Those dendritic cells get activated, and they present antigens to T cells that become cytotoxic. Those T cells should then identify cancer cells and eliminate them. There are, however, many resistance mechanisms, as we know, and cancer cells, for example, can express PD-L1 that will prevent this from happening. So we looked also at combinations with the checkpoint blockade therapy, here anti-PD-1, to see how this can potentially synergize with the STING agonist delivery. STING agonist can enhance and prime T cells, but also stimulate natural killer cells, as well as polarize macrophages to become, instead of M2, to become M1 phenotype that is anti-tumorigenic. So it actually communicates with many of the immune cells in our system to form an anti-tumor response. So nucleic acids, as we know, the STING agonist, RNAi, microRNA, or plasmids, are rapidly cleared from the system, and their negative charge really impedes their ability to enter cells. So for a lot of those, we use nanoparticles. And as I said, polybeta amino esters that I mentioned before is a great polymer because it's biocompatible. It has functional groups that can help us with modifications and conjugations of therapies. And it has good buffering capacity that enables it, after uptake into the cell, to escape to the endosome and go to the cytoplasm. The problem is that when we think about STING agonist, it's really at the lower spectrum of molecular weight, and hence the electrostatic interactions that are usually formed between nucleic acid and the polybeta amino esters are too low to make it stable and amenable for systemic delivery. So here what we had to do actually is, for the first time, conjugate the STING agonist or the CDN molecule reversibly to the PBE nanoparticle via a cathepsin-sensitive linker, which means once it goes into the cell, it will cleave and release the CDN. And with that, we got very effective results. Here you can see in a melanoma model, when we apply the CDN systemically using the nanoparticle, we see very nice attenuation of tumor growth, while the free CDN alone did not make an effect even compared to untreated mice because of its rapid degradation in the serum and inability to enter cells. This results in improved survival. But when we combine it with a checkpoint blockade, anti-PD-1, you can see an even greater attenuation of tumor growth that resulted now in 60% curation of the mice. We got very positive results also in colon cancer, as well as extension of survival in triple-negative breast cancer. So this really shows us the ability to deliver very potent nucleic acids, even those that are very small and that conventionally will be very hard to deliver by electrostatic interactions only, using a more sophisticated reversible conjugated methods that can deliver the cargo to wherever it needs to go. So to summarize, we've seen that non-viral delivery systems are really promising. We can deliver a range of cargoes using different types of nanoparticles, and we really have a very large armamentarium of materials that we can use for delivery depending on the cargo and the target site. Many new formulations are entering clinical trials, and future iterations really need to focus on understanding nano-bio interactions in order to design safer and more potent therapeutics for a range of diseases. So in the past 2 years, with the COVID-19 vaccines from Moderna and BioNTech, we've seen how non-viral delivery systems, when combined with the right therapy, can actually change the face of a global pandemic. And we believe that with the right delivery vehicles and choice of nucleic acid, we can have the same transformative outcomes in terms of the treatment and prevention of many other types of diseases. Thank you.
Okay. So if it's not evident by the fact that we're both sitting down now, we're going to move to the Q&A session. If you have any questions, please raise your hand. The purpose of this is to be interactive, and that's a benefit of all being here together in person. So maybe just to start, though, very, very interesting talk, Natalie.
Thank you.
And you mentioned a few different benefits that LNPs can have over the more traditional viral-based vectors. But I guess, in terms of the targeting specific tissues, specifically, how does this really overcome the traditional limitations of a viral-based approach?
Thank you. So I think the most striking difference between non-viral and viral delivery systems is the ability to rationally design the platform in the former versus dealing with what nature gives us. Now, in some cases, viral delivery systems can work really well for the target and the indication of interest. But with non-viral delivery systems, we have a much broader landscape of types of materials, formulations, and modifications that we can use to get what we want. So the idea would be to actually use a large screening, high throughput screening of materials, and to start to understand how different formulations and different determinants control the outcomes that we are seeing in terms of the mode of action, the uptake into cells, the distribution, toxicity. So this is really enabled by a non-viral delivery system that we can really modify conveniently. The other aspect is that a lot of those nanoparticles are amenable to different administration modes. So as I mentioned in my talk, we can deliver, for example, for cancer therapy, we can deliver intratumorally, we can deliver systemically, we can deliver in a prolonged manner locally or transdermally, really leveraging the idea that in the skin, for example, or muscle, we have a lot of immune cells that we can tap into, especially with immune therapies. So the ability to modify the particles, to identify how their structure affects their function and performance, and also control where and how we deliver them can give us a lot of freedom to operate.
Right, right. And I mean, in terms of the hepatic delivery, right, ApoE plays a significant role here. Could you just talk about that?
Yes, so we can also leverage the underlying biology. So for lipid nanoparticles, ApoE really enables their efficacy. We can also rely on overexpression of receptors to better target. And we can even think of biomimetic materials where we can use viral capsid-derived proteins to modify our non-viral delivery systems, really getting the best of both worlds. So there's a lot of room here, a lot of creativity, and a lot of room to engineer our vehicles.
And I guess what is sort of the carrying capacity in terms of the targeting proteins, whether it's just a short protein or something as large as maybe an antibody? Is that also feasible? for targeting?
Yes, absolutely. So we can actually modify the nanoparticles with different targeting agents. It can be a peptide, it can be an antibody to really modify what cell it will go into. We can also use different selective molecules that will change the physical and chemical properties of the particles that will drive what tissue they go to. So we have a lot of control over the tissue site and the cell types that the particles will go into.
Right, and like your presentation is not just the way that the vector is designed, it's also the vehicle, I guess, that you're delivering the vector in, whether it's a hydrogel or something like that.
Exactly.
And I think this question about selective targeting to tissues is important from a safety perspective as well. We've seen with some of the more traditional, adenovirus-based approaches, sometimes there's innate immunogenicity against those viral particles that can either mute the response or can lead to sort of systemic effects, which obviously aren't great. And I think the recent FDA advisory panel on AAV-based gene therapy, that was a major component, right, which was the safety aspect. So in your view, I guess when we're thinking about LNPs, what needs to be done to ensure that they're safe? And I guess what are the benefits of LNPs over AAVs in terms of safety?
Yeah, so I think with the recent COVID-19 pandemic, we've seen that in general, LNPs are safe. And of course, we've seen also variant responses between people, and we need to understand what drives that. But in general, for materials, we need to, again, screen a large amount of formulations in order to understand how their structure affects the properties, efficacy, and toxicity. But you also need to think about the cargo. So what are we actually delivering would also make a difference, right? So its mode of action, its immunogenicity, the dosing regimen, the dose that we are delivering, and where it goes will really affect the outcomes. The other aspect that we need to consider is how the preclinical models that we're using really recapitulate what we see in patients. In fact, there's been a recent publication that showed that in mice, their response to the LNP mRNA is much different than in humans. In fact, there's interleukin-2. Actually, patients are sensitive to 1,000 times less amount of IL-2 compared to mice. So the cytokine storms that we've been seeing in patients, it was hard to really anticipate based on the studies in mice. So again, high throughput, ability to work with larger models. Non-human primates would be great, but of course we cannot screen in non-human primates, or not different types of mice that are modified to represent aspects of the immune system of patients. So the idea would be potentially to use a more sophisticated ex vivo model, so 3D multicellular models, as well as organ on a chip to be able to really screen for different materials and drugs in the context of immune cells that represent what we'll see in patients.
Right, and I think that was a major component also of the FDA advisory panel last year, which was how do we design our preclinical models to better predict for these safety outcomes. So, what are the advancements sort of being made there from the non-viral based vectors?
Yeah, so here again, we can leverage the idea that we can modify and engineer those particles such that we can see what components may drive toxicity, for example. So again, large screening, a lot of data, and potentially AI can help us identify these formulations that are safe.
Interesting. And you mentioned the mRNA LNPs, and obviously that's the topic du jour given the success in COVID, which was really, I think, the proving grounds for non-viral nanoparticles as a means to activate the immune system. So I guess, can we take the technology developed for COVID and apply it to, indications, outside of infectious disease like cancer or even cystic fibrosis, something like that?
Yes, absolutely. So I think that's why it's so exciting. We have a delivery system whose safety has been established, and we can now plug and play with the type of nucleic acid or therapy that we are delivering. So in cancer, in fact, some of the clinical trials with mRNA predated the COVID-19 pandemic. So the idea is to think about prophylactic versus therapeutic setting of cancer, how nucleic acid delivery can potentially synergize with some of the standard of care therapies like chemotherapy and radiation, how the timing and dosing regimen affect the outcomes that we're seeing. So really the idea is to use new and emerging therapies along with existing therapies to get better outcomes. And we can use it not only for cancer, as you said, it can be also for autoimmune diseases. We can deliver nucleic acids that will suppress the immune system, such as skin transplants, multiple sclerosis, et cetera, to prevent rejection. So it actually opens up a lot of opportunities to treat a range of diseases.
And you mentioned STING in your slides. How does that sort of, how could that be one angle to sort of?
Yeah, STING is an interesting molecule. As we think about targeting and where we want to deliver the nanoparticles, in fact, in STING, as I presented, it activates dendritic cells, macrophages, natural killer cells, T cells, all of which we are interested in targeting because they all synergistically eliminate the cancer. And when we deliver the STING agonist nanoparticles systemically, we know that cancer cells will be also major recipients of the particles because there are millions of cancer cells in the environment. And in fact, a lot of the particles that we are now using to target immune cells were originally developed to target cancer cells. But we know, based on experiments that we've done with knockout cells, that cancer cells are not responsible for the effect that we are seeing. So they're not being activated by the STING agonist delivery. So the question is, how targeted do we want to be? We want to actually get the STING into many of the immune cells. So maybe what we want to do is actually block them from entering into cancer cells. So it really depends what we are delivering. There are some therapies that, if they go to cancer cells, may actually antagonize the therapy, the immune therapy of interest. So we really need to think about what the target is, what the mechanism of action is when we design those nanoparticles, and think when we need what type of molecules we can add to enhance the selectivity towards one cell or the other.
Right. And it's not just the selectivity component of that. It's what can you package, right, inside the LNP. So could you theoretically put, I don't know, like a Keytruda or an antibody therapy inside of an LNP?
Yes, so actually you can have a lot of different types of therapies inside the liposomes, but there are many other types of particles that can be used as well. Sometimes you want your therapy to actually be exposed, to be on the surface of a particle, and that's why dendrimer or polybeta amino esters, some of the other polymeric particles can be very effective. So especially as we think about combination therapies, you can think about particles that will encapsulate some of the drugs or have on their surface conjugation of the other drugs. And this enables also to decide what is exposed and what is released afterwards. So you can play with a lot of different types of particles as you start to think about combination therapies.
Mm-hmm. Mm-hmm. And can you talk about the formulation of LNPs? What's sort of the flexibility there? I mean, intramuscular has been sort of the go-to for at least the vaccines. So what's the flexibility to go intravenous or even topical or inhaled through a nebulizer or something like that? Yeah,
so they can be delivered using a range of approaches. We have some freedom in terms of the formulation. It's used from many different types of lipids, and the type of ionizable lipid really determines where it will go. But we can also use that along with other biomaterials, as I presented, either hydrogels or needles or a depot that can be applied intranasally, for example, to control where it would go. So we're not only limited by the actual type of particle and the freedom that it provides us with, but also these can be combined with other delivery methods based on other biomaterials to get the particles to wherever we go in the regimen that we want, the order that we want, to get the outcomes that we need.
Right, right. And I thought your combo data in your slides with chemotherapy in mice was particularly compelling. I guess when you look at combining with standard of care, what would sort of be an ideal formulation or does it depend on the tumor type?
Yeah, so in fact, it's very interesting that there can be synergistic effects between new and emerging immune therapy drugs and clinically available modalities. For example, a lot of the chemotherapy drugs and radiation therapies are known to kill cancer cells in a way that they release their content. So now you're exposing a lot of antigens. And when your immunotherapy comes, take, for example, the STING agonist, what STING does is actually activates dendritic cells to present antigens to T cells. So the ability to expose those antigens should synergize with the STING delivery. So a lot of the chemotherapy drugs and radiation can help with that. We can also use nucleic acid sequences that will sensitize the tumor to radiation and chemotherapy. So to really make those drugs work in concert with each other to get better outcomes. So there's a lot that we can do, actually, as we think about combination therapies, leveraging the biology and the standard of care therapies that are already available. And this is actually important not only because we can improve the therapy, but in fact to minimize unpredicted outcomes. For example, when we go and treat patients in clinical trials, in many cases it will be end-stage patients that have already received chemotherapy and radiation therapy, and we really need to understand how this changes the gene expression of those patients, epigenetics, and the microenvironment in general as we deliver our new and emerging drug. Without understanding those mechanisms, there can be failure. So it's important for a variety of reasons to understand how we can combine different therapies and how they may work with the standard of care modalities.
And along those lines, what's being done in the preclinical setting in terms of, you mentioned immunotherapy plus chemo. Are there other sort of combinations that either you're working with or you've seen in the literature? And I guess what sort of preclinical models are being used to test these?
Yeah, so with a lot of the immune therapies, now we need to use immune viable mice, so not immune compromised mice as we used before, because we want to see how the immune system is actually responding. So now we try to use a lot of those models using different cell lines that have different responses or sensitivity to immune therapies to try and understand how a heterogeneous tumor will respond to a therapy. We also use patient-derived xenografts, so tumors from patients. The problem with those is that you need to use immune-compromised mice. So while this can show you how your therapy can potentially deal with a very heterogeneous environment, you cannot learn from that how the immune response would look like. So really the idea is to use multiple models, multiple cell lines, and as I mentioned, also ex vivo models, and all of them together will give us a better idea as to what the outcome would look like.
Right. I guess, do you see a role, obviously in the context of infectious disease, right? You're trying to stimulate the immune system using an antigen. The same applies to cancer, so to speak, but some of these patients maybe don't have an active immune system. In the case of, leukemia or lymphoma. I guess, what do you see in terms of the immune stimulating effect in that context? And I guess would sort of like an adjuvant or a neoadjuvant be required?
Yeah, so here we can think about neoadjuvant therapies to activate the immune system, to get more cells migrating to the site of interest. We can also think about combination with cell therapies, CAR T cell therapies or nucleic acids that can be injected to directly modify cells in our bodies. So we have now a lot of potential opportunities, but also a lot of challenges with those. So there's a lot of room for research to understand how we can better leverage everything that's available for us in the toolkit to get better outcomes in a real scenario in patients.
And this is just me thinking out loud, but is there a way to sort of incorporate a neoadjuvant in the, you mentioned hydrogels or something like that. Would that be a safe approach to sort of getting it all in one?
Yeah, that's definitely one approach. The question is if we know what the neoadjuvants are for each tumor type. When you know what it is, then it's an excellent approach. In many cases, you either don't know or there are multiple actually antigens that you need actually to expose. So in this case, actually, eliminating the cancer cells, releasing those antigens in situ, and then delivering your therapy can cause in situ vaccination where your antigens will be right there and you don't necessarily need to deliver them. So it really depends on the, how much knowledge we have on each tumor.
Right. And we've talked about mRNA, we've talked about protein. How about other sorts of therapeutic modalities, whether it's RNAi? What are sort of the considerations, when you're trying to knock down the genome?
Yeah, so we know now that one pathway is never enough, or monotherapy is never enough. So when there is a particular gene that is mutated and we can actually edit that, sometimes it can really transform outcomes. And in some cases, it is not enough, and you need other modalities as well. So I think the fact that we can utilize many types of nucleic acids, many types of therapies that can work together will probably be the solution moving forward. But we can think about protein replacement therapy. We can think about editing with CRISPR for a range of diseases. So we can think about infectious diseases as well, HIV and others. So there's a lot that we can do, but we'll have to combine many different types of modalities in most cases.
Right. And it does, like you said, it depends on the patient history.
Exactly.
So I guess how does, how could one day, an LNP-based gene therapy for cancer be tailored to specific patients, or is that even feasible?
Yeah, so personalized medicine is an interesting concept, and in some cases it may work really well. In other cases, like for example in glioblastoma, where the tumor is very heterogeneous and there's a large variability between patients and patient history in terms of radiation and chemotherapy, and then alterations of the genes that really make it a bit hard to make it personalized. But really the idea is to potentially one day leverage materials that can respond to their environment. So you can imagine delivering nanoparticles with many different types of therapies that will get activated only based on signal that exists. So you can have actually a whole pharmacy on a chip, and it will get activated for each patient based on their need. And that will be cool.
Oh, that's very interesting. And it sounds like you're doing some of that high throughput work in your lab already, right?
Yes.
So maybe a few years away from that being in actuality, but
absolutely.
Yeah, steps in the right direction. So I guess, just taking a step back, you mentioned, sometimes tailoring the material based on, the disease or the individual patient, whether they have a skin metastasis or something like that. So I guess, how do you choose the right material for the job from the toolbox?
Yeah, that's not easy either. There are many considerations. So you can think about the type of material, what its natural tropism, physical chemical properties. You can think about the cargo, what you're actually delivering, what's the mechanism of action, where it needs to go. And you can think about the context or the microenvironment, what therapies the patient has already received, even the different administration routes, the dosing regimen, and the different combinations. So all of that need to be taken into account. Sometimes many different particles can do the same job, and sometimes you need specialized particles to do what you need. So I think the idea is, again, we have a toolbox we can choose from. In many cases, when we deliver a new technology for delivery of an emerging therapy, there will be some screening associated with that, informed screening, based on the knowledge that we already have. And then we'll identify, leading candidates, and then start to understand how the stability of the particle and distribution is affected by administration, by timing, by combinations, see where it goes in terms of the organ and even cells. We do all of this analysis and then identify the lead candidate that then we continue to do all the work with. So again, it's good to have options and then screening capacity to get to the leading candidate.
Right. And I guess, could you just speak to sort of the intrinsic versus extrinsic properties of the different materials that are being used, and how this might be tested sort of in vivo?
That's a good point. So the intrinsic properties of the particles will be leveraged, and we'll see how physical chemical properties affect, again, distribution, uptake, and outcomes. But sometimes we can use materials that can respond to an external stimuli. So for example, we can use light or ultrasound to activate particles only where and when needed. So for that, we can think about particles such as gold, for example, that I've demonstrated. The gold with the near-IR laser, for example, can heat up. So for cancer therapy, it can kill cancer cells, expose antigens, and then your therapy, your emerging immune therapy, let's say, can act when the antigens are exposed. So this is another consideration. When we want the materials to work with an external stimulus, then it will also, direct us towards one material and not the other.
Very interesting. And I guess when you think about translating to the clinic, right, it sounds like there's going to be a lot of work that goes into particular therapies up front in the lab. I guess, what is sort of the preclinical work that will prepare these therapies to eventually go into humans, whether it's the safety aspect, whether it's the designing the material for the indication?
So I think one major question always, and a debate as we develop new particles, is do we actually go to those materials that are already FDA approved and it will be easy to use them? And the go-to was PLGA and now liposomes. Or do we actually try and work with new particles that will have to get approved and go through all of these? But in some cases, it may be needed, and that's what will make the difference eventually. So that's one aspect that we need to think about. And of course, if there's something that's approved and simple, we can go to that. But sometimes you need to come up with new solutions. What we do is we use preclinical models. We start with mice, because with mice, you can image everything very easily. You can see where your materials are going, where the drug is going, and you can see the response to the therapy using luminescent and fluorescent signals. You can understand mechanisms. There are many models that are available. But then once we optimize the therapy, we go usually to larger animals, sometimes rabbits or pigs, depends on the model, and then to canine models or non-human primates, again, depending on what we're delivering and what the disease is. And usually this data is required for FDA submission. The other consideration is, again, is the manufacturing of the material. So that's the benefit of going into an FDA-approved material where the manufacturing already exists. You can scale up your delivery system with the drug, and with the GMP material, repeat under GLP conditions some of the key preclinical experiments, and then submit that to the FDA.
Interesting. And one trend that we've sort of seen with the mRNA-based vaccines for COVID is, originally people thought you de-risk the LNP, you de-risk the technology, mRNA, and then it's just modifying the sequence, and maybe you run another, small phase 3 and it can get approved. But when they're applying this to flu now, it doesn't seem like that's really the case from a regulatory perspective. They still have to step through the phase 1, phase 2, phase 3. I guess when you look at the LNP technology itself, would one need to design a clinical study each time, or can you maybe get a large enough safety database for a particular technology where maybe it's a little bit more straightforward?
Ideally, we would be able to apply for accelerated approval, so maybe lower number of patients, less time. But in essence, we know that with anything that we deliver, the cargo will make a difference. The immunogenicity of what we are delivering, the different antigens, the different drugs, their potential toxicity is different. So we're talking about people, so I suspect that we'll always need to go through all the phases, unless we're talking about emergency use or we are talking about late stage diseases like cancer that there's no other alternative and there's a few months maybe remaining for patients to survive, and then you can probably get more accelerated approval. But I suspect that since there's a lot of unknowns, we'll have to go through the chains.
Right, and especially, you mentioned cancer, even within a particular indication, not each cancer patient is the same, right? So
exactly. It's
not like you could just, de-risk it in melanoma and apply it directly to lung cancer, right? Okay. I'm going to pause for a second and see if there's any burning questions. Yes?
Thank you so much for amazingly graphic, inspiring Chemistry, materials, and biology, and disease and treatment. A question about translation of biodistribution without taking into account the cargo and all the biological differences between mouse, pig, non-human primates, and humans. Is there a more reliable biodistribution translation between small and larger animals in your preclinical models, in your experience with these particles?
So just to repeat the question is there direct translatability between small and large animals, or is there some disconnect between those models?
Yeah, so there is some disconnect. As we know, for example, for cancer therapy with nanoparticles, in small animals, we really relied on the EPR effect, so enhanced permeation and retention of particles in the tumor microenvironment because of the leaky vasculature. And it's really up for debate whether this mechanism even exists in patients. So while we were relying on a very passive mechanism of accumulation, it may be that actually active targeting is necessary, although again, it depends what kinds of targeting and how much this can enhance accumulation and how this affects biodistribution. And as we think about biodistribution, yes, we can learn a lot in terms of how the different functionalities and structures of the material and their physical chemical properties affect distribution. But once we add a cargo, this also affects the properties of the particle. So we can learn a lot, but there's always going to be the need to study very carefully the final product and go into larger animal models with the idea that it will be closer to what was seen in patients.
So, thank you very much. It was very illuminating talk. My question is a slight variation of what she asked. So this one example you gave about microgel, and you showed a tumor in the brain was removed, and then you applied the gel. Same question I have, the biodistribution. How you will make sure that whatever cargo you are giving will be distributed in the rest of the brain, if that's your aim?
Mm-hmm.
Right. So the question was, how do you formulate the material to really constrain, the delivery of the drug, depending on the tissue?
Yeah, so in the brain, it's very interesting. Glioblastoma, for example, the tumor grows and it spreads. It has tentacles that you actually need to reach, because even with tumor resection, that 70% of the patients will undergo. For the other 30, it's unresectable tumor. But even with tumor resection, you cannot remove all the cancer cells. So you actually need your drug to be right there and be able to diffuse and penetrate the tissue following the resection in order to eliminate those cancer cells. Now, this is a very aggressive and infiltrative tumor. So you may have cancer cells in the other hemisphere, and we cannot expect that the drug will diffuse all the way there. So that's why combination with immune therapies that will train the immune system to identify the cancer cells and eliminate them, because then the immune cells are circulating in our body, can be the solution, together with chemotherapy and other therapies that will control the tumor growth, can be the answer. So we need a drug to be at the site based on what we've seen, because this allows you to overcome the blood-brain barrier, but we need it to also be retained there and be released locally over time. Otherwise, it's going to be eliminated very quickly before you can see an outcome. And when you do that, you probably also need to train the immune system to make sure that everything will be eliminated.
I think we have time for one more question.
Maybe two, because I just want to follow up on that. But very good presentation. Follow up on that one. So what's the outlook for being able to use a lipid nanoparticle to cross the blood-brain to use systemic circulation to get into the brain? So you do get dissemination to various tissues. So that's the follow-on question. The original question was, can you compare this to exosomes, a natural vesicle, lipid vesicle, and the advantages and disadvantages?
So two questions. Can an LNP be designed to cross the blood-brain barrier from circulation? And second is, what are the similarities or differences between a man-made lipid particle and a pre-existing one in nature?
Yeah. So whether LNP or actually, in fact, any nanoparticle can cross the blood-brain barrier, it can to a certain extent, and we can modify the particles to do so. However, in our experience and based on publications, it usually is not sufficient. So we have many different ways to enhance the accumulation and penetration in the brain, using blood-brain barrier disruption methods, for example, using focused ultrasound and different modes of transcytosis and penetration. But again, the question is if this will be sufficient. So ideally, you can potentially combine a strategy like that with a local delivery platform that then together can work in concert to eliminate the tumors more effectively. So yes, we can engineer the particles to penetrate. The question is how much and retention. So as I've demonstrated, yes, you can penetrate, but even when we injected drugs directly to the tumor in a free form, we didn't see effect. Now, with regards to other lipid formulations like exosomes or extracellular vesicles, the benefit of those, they are naturally designed to transfer information between cells and have natural tropism to a certain cell, and we can potentially leverage that. But the flip side of that is that it's very heterogeneous. It's hard to make a product based on that, and there's still a lot to learn before we can actually use that. So absolutely another tool in our toolbox, but there's a lot that needs to be discovered still. And perhaps we can use their natural capacities with some of the engineering tools that we have, and again, use hybrid materials. But again, there's a lot of opportunities, and that's why it's so exciting.
Great. Well, that's it for time. Thank you for your questions, and thank you, Natalie, for the thoughtful discussion. Really appreciate it.
Thank you for your question.